start today's class. Okay, so in the previous class, we had discussed about how the biotechnology has its application in the field of medicine, in which we were discussing about the insulin which has been genetically engineered. We saw that the insulin is made synthesized and secreted by the pancreas, particularly the beta cells of the pancreas. And we know that the insulin lowers the blood sugar level. Now, a deficiency of insulin, it causes a disease that is known as diabetes mellitus. So, if a person does not have enough insulin in the body being made, so that person will have diabetes mellitus. So, if a person wants to treat this disease, they have to take periodic injections of insulin. Now, earlier, like I told you, that these injections, or you can say the insulin that were present in the injections, they were extracted from the pancreas of the slaughtered cattle and pigs, from their pancreas, because they are very similar to humans, like their genes are very similar. But this created a very big problem in the market, because first of all, it was like the people that were using it, they had several sorts of, you can say, allergic reaction to these animal extracted insulin hormones. Apart from that, ethical, or you can say religious, sentimental beliefs were also mentioned, or you can say associated with it. So no one wanted to do this. Now then came this company called as Eli Lilly. Now this Eli Lilly company, it was like it thought to create artificially the insulin that is required. Now I asked you all, why does the insulin have to be injected?
Because if we eat it in tabular form, our stomach will burn.
Our stomach will
Degraded.
Degraded.
degrade it. So why does the insulin have to be taken through injection? So the answer to this is that oral administration, you people understand, taking something orally, administration means taking it. So oral administration of insulin can degrade it in the alimentary. Do you people know what alimentary canal is? What is alimentary canal? It will degrade it in the alimentary canal. Okay, this is clear. So, so that is the reason why the insulin injections have to be taken. Now, what happens in, like, if we talk about the structure of insulin in the humans? Okay, so insulin is made up of 3 chains actually. In the case of humans, A, B, and C chains. Okay, and each of those chains are sort of joined by the disulfide bonds. Disulfide bonds, you people understand? Disulfide bond. What is the meaning of it? Disulfide, tell me the meaning by the name only.
Two sulfide.
Two sulfides are there. Disulfide, students. I'm not asking you the chemistry of it. You know it better than me. But I was just asking, what is the meaning of disulfide? Meaning 2 sulfate bridges are there, which joins each of the chains of this thing, insulin together. Okay, so what happens is that normally There is chain A of insulin, chain B of insulin, and chain C of insulin. Now, in humans, the insulin has 3 chains. Okay, now see, whenever I say that they have 3 chains, remember that this 3 chain structure, wherein they have chain A, chain B, and C. So altogether, it is an inactive, or you can say pro-hormone. Pro-hormone, you understand that to become fully mature, it has to lose 1 chain, meaning it has to undergo some sort of modification to become, you can say, active, or you can say become a proper functioning hormone. So we can say that insulin in humans, insulin is synthesized as a prohormone. As a prohormone. Also, students, I've been meaning to ask you all, are you people facing any difficulty in the assignments or assessments that you are given after the class?
No, ma'am.
Hmm?
No.
No? How many of you make that assignment or do that assignment, and how many of you just like toss it out? Tell me honestly. I'll not judge you. Maybe a little, but not a lot. How many of you genuinely take it seriously to do that assignment, and how many of like, oh, toss it out? Give your honest opinion. I am not going to judge you.
Mm,
I'll only judge you this much.
I somewhat take it seriously.
Somewhat. Okay. What about others?
Like
He
if I,
said the truth. He doesn't take it seriously, so he said the truth. What about others? Do you take it seriously or no? No, not even a bit. Not even the amount I was going to judge you at. Not even that much.
I want to do it, but like I
Huh?
just don't get time,
You don't get
and
the time.
and then
You
I
don't get
forget
the time to
about it.
study. Okay, this was question number 1. The second question is that how, like, what is the time that you people devote to your self-study? Have I asked this question before?
no.
No, okay. Because I was going to use a different dialogue if I had asked before. But since I'm asking for the first time, tell me. And my
Like
girls, all of you have changed your look. You people look so different. Okay. Tell me. Coming back to the topic.
how many hours I study or something.
Self-study, not me teaching you, not your school teachers teaching you, not anyone teaching you, teaching yourself. Self-studying, wherein you learn things, wherein you understand. How many of you don't do it at all? You're like, let the exam come one day before, I'll do everything like a marathon. Why should I take
No,
tension beforehand?
no, I don't do that.
You don't do that?
Yeah, I dedicate, for example, after tuition, like from 9 o'clock here till 11.
9 to 11.
Yeah,
Okay, what about the others?
3 to 4 hours.
3 to 4 hours. Genuine study, or the one wherein you read one
Yes,
line,
yes,
some
genuine.
Instagram, read another line, some YouTube.
No, no, genuine.
Genuine study. Okay, Mariam, what about you?
In my school, we have assessments every Sunday, Tuesday, Thursday, like
Hmm.
every week. So like
Hmm.
I only study for that, and that's enough.
That's
Because
enough.
yeah, like all the chapters they cover, like I have 20 marks assessment every Sunday, Tuesday, Thursday.
And so all of your chapters get covered in that.
Yeah, yeah.
Okay. You people know how much marks I expect from you all, right?
yes, ma'am, 60 above.
Eh? 60
70,
above?
yeah, 70 marks paper, no? So 60 above.
Oh, okay. 70 marks paper is there. Okay. Of course. I thought out of 160 above. Yes. definitely 60 above, but I expect full marks from all of you, at least in biology. Because I teach you all with my heart and my soul and every possible fiber of my being. So I need that return. I'm telling you all. Be serious with your studies, okay? And do as much as self-study you can. Oh God. Yes. As much self-study you can do that. Okay. So yeah, so we were talking about the insulin. So the insulin is made up of 3 chains, A, B, and C. But always remember that insulin, when it is synthesized and secreted out of our body, it is a prohormone, meaning it is sort of inactive. For it to become fully, you can say, active, it has to be processed. Okay, processing of the insulin hormone is very, you can say, necessary. And when it gets processed, it becomes mature. Now, if the mature insulin is present in our body, it will do its function. That is, what is the function of insulin in our body, students?
To lower the sugar in our body.
To
Lower the
Sugar,
sugar.
to lower the blood sugar level. That is the function of insulin. Very good. Now, see, the prohormone that is synthesized, that has an additional chain called as the C chain. That is the additional chain. So, for the insulin hormone to become a mature hormone, that C peptide or the C chain has to be removed. Okay. So, to become mature, students, after this chapter, meaning biotechnological applications, what other chapters do you people have left? Check in your list. Tell me. How many chapters do we still have to cover?
I have 2 chapters left, ma'am, after this biotechnology. Organisms and population, biodiversity and conservation.
Okay. Same for all, right? Because the syllabus is the same.
Yes, ma'am.
Environmental issues are removed. That chapter is removed, isn't it? It's not there for you. Okay, so only 2 chapters.
Yes, ma'am.
All right. So I can take my time to teach you. Now I don't have to be in a rush.
Did we do ecosystem?
Oh, ecosystem must be there too.
Yeah, we have.
Yeah, organism population, diversity, and ecosystem. 3 chapters.
Organism and population, we did that, right?
Have we done it?
Yeah, we did that.
The
Before biotechnology, we did that.
mutualism, proto cooperation, all of these things we have done.
Yeah.
Okay,
You said
so
it was EVS and stuff.
yeah, it is indeed EVS.
Yeah,
So
ma'am.
we
We
have
also have
EVS chapters,
ecosystem.
ecosystem and
Ecosystem,
biodiversity.
biodiversity. 3 chapters
Yeah.
left after this.
Okay, so I think majority of the syllabus has been covered, isn't it?
Yeah. Yeah.
Yes, ma'am.
12th is like shorter.
Yes.
So, so to make the insulin a mature insulin, we have to remove the C chain. Okay. Now, what used to happen is that this C chain removal was a difficult process because normally it takes place in our body naturally, but to do it on, like you can say, artificially or on an industrial scale, it was very difficult. So what the genetic engineers thought, that we will synthesize the insulin that does not even have this whole entire complication of having a third chain. So they synthesize insulin with A chain and B chain. That's it. Okay, and to see that procedure, I'll show you this picture. Okay, see this. Now, the plasmid is present inside the E. coli, which is the bacteria, right? And plasmid, we know, is a carrier, right? It carries the gene to the host and all of that. Now, the human pancreatic cell, it synthesizes the genes for the insulin. So the gene of interest comes from the human cell, obviously. So from a healthy human cell, the insulin gene is extracted with the help of restriction enzymes. And with that same restriction enzyme, the plasmid is also cut. Now, the plasmid, since it has been cut, it has sticky ends. What are the two types of end that is developed, students? One is sticky ends. What is the name of the other end? Hmm? Blunt ends. Remember that? Blunt ends. So ultimately what happens is that we take the cut plasmid and we take the gene of interest that has been cut and we join them together. We like stick them with the help of the molecular glue, that is DNA ligase. And this process can also be called as annealing. Now this is the recombinant DNA, obviously. So now this recombinant DNA is introduced inside the bacterial cell, and the bacterial cell, with its own multiplication, with its own cell division, it synthesizes a lot of insulin molecules just like that, because it has been incorporated in the gene of it. Now the clones of engineered bacteria in a perimeter containing medium. So now the clones, meaning multiple copies of that particular bacteria is made, and then the insulin hormone is extracted from them. So this is how the insulin is made. Here there is no tension of, you can say, creating or removing a C peptide chain or making the insulin mature or any of that sort. Is it clear? So chain A of the insulin is produced separately, chain B is produced separately, and later on they are joined with the help of disulfide bonds. Okay, so I'll write that also, that chains A and B were produced separately and then they were extracted, right? Once you do this procedure for chain A, secondly, you do the procedure for chain B. And then they were combined. And then they were combined by creating sulfide bonds, by creating disulfide bonds to form human insulin. Is it clear, students? And which company did I say does this or did this? What is the name of the company that I took? No one listens to me these days. So sad.
No, I just
Tell me.
Eileen something.
Hmm?
Eileen, I don't know.
You should remember the year also. In 1983, the company's name is Eli Lilly. Okay, Eli and Lilly. So Eli Lilly company that created, it was an American company. Okay, it is a company based in America. So this company synthesized two chains of, you can say two DNA sequences of insulin and later on joined them. So Eli Lilly, an American company. An American company prepared two DNA sequences. Okay, so it prepared two DNA sequences, that is A and B, and later on the whole entire process was repeated just like that, meaning they joined it together, they created the mature, you can say, insulin. Okay, now tell me, which is one of the most common vector, the name of the most common vector? Hmm? So this is like very basic questions that I asked. It's not even a tough question.
E. coli.
Hmm?
E. coli.
Okay, E. coli, correct. But E. coli is a bacteria, it's not a vector. Which,
So that PBR, that
PBR 322 is the name of the vector. Very good. PBR 320. Always remember, this is one of the most studied, most common vector. And this is actually a plasmid, right? We know that plasmids are vectors. And this plasmid is of E. coli. E. coli is the right answer, but E. coli is not a vector. It's a bacteria, isn't it? It is the most studied bacteria for anything. You take the lacoperon, you take anything. It's like everything when it's related to the bacteria, it is studied on E. coli. Okay. So same way, E. coli's plasmid is one of the most useful plasmid because it acts like a vector during the RD. And the name of the plasmid is PBR 322. Now you remember I showed you the structure of PBR 322 wherein it had antibiotic resistant sites. That can directly be asked. Where like at PVU which resistance is present, at PST which resistance is present. So you should learn that diagram, practice that diagram as much as possible. Okay, maybe I'll not ask it. Maybe I will, but in the exam also it will be asked. So beware about that. Okay. Now, the next thing is gene therapy. Now, what happens is that the biotechnology, it can also be used to correct the genetic disorders much before it's too late for the human being to survive. Okay, so what happens? It provides the patient with a correct gene that it requires because many a times, because of mutation, there are many people that can lack a proper gene, that cannot have a very good expressive gene. So those can be replaced by gene therapy. And the field of biotechnology helps us to do so. It helps us to, you can say, cure the genetic disorders. Okay. So first you people note down till here, then I'll move forward. So wherever the diagram is there, you note that diagram. It's like very easy to learn from that diagram.
better, ma'am.
And ma'am.
Ma'am, do we have to draw the diagram as well?
Yes, the whole thing, so that it'll be very clear to you, okay?
Okay. Done, ma'am. Done, ma'am.
Everyone done?
Yes, ma'am. Ma'am, 1 minute actually. 1 minute.
Okay, okay. Take your time, it's all right.
Done.
Done.
Done, ma'am.
Okay, any doubts up until now, students?
No, ma'am.
No, ma'am.
No, ma'am.
The whole entire production of insulin, why is it needed, where is it needed, how is it made, all of these things are clear?
Yes, ma'am.
Okay, very good. Now the next thing that we are going to see is gene therapy. Why is it done? When it is required to be done, and all of that things that come along. Okay, let me just. All right. Now this is a very descriptive paragraph. This is like basically all we need to know as far as the gene therapy is concerned. Now, like I told you, there are many human beings that have some sort of inherited disease. Inherited diseases, you people have already studied this, right, in the human health and disease. There are certain genetic disorders that a person can be born with. Now, the particular disease can be corrected if a proper gene therapy is done. Now, the gene therapy, this is the name originally given to the methods that aim to cure an inherited disease. Can you name some inherited disease, students? Anyone?
Sorry, ma'am, could you repeat?
Color blindness,
Color blindness, then
hemophilus,
Sickle cell anemia,
sickle cell anemia is there, hemophilia is there,
cystic fibrosis,
yeah, cystic fibrosis is there. There are so many diseases that are inherited. So, gene therapy is that method that aims to cure the inherited disease. Because normally people think that, okay, a person has got this disease from, you can say, the mother, and it has been inherited. So, we think that it cannot be cured at all, isn't it? That's what our thinking about is. Now, how is it done? It is done by providing the patient with a correct copy of the defective gene. For example, let's just say like in sickle cell anemia, the uh, glutamate was replaced by valine in the sixth position of the beta globin chain. So maybe the glutamate was given. So a correct copy of the defective gene, whatever gene has been defective, because of which the disease is produced, that a proper, you can say, version of it or the correct version of it is provided to the patient. Now, gene therapy has now been extended to include attempts to cure any disease by introduction of a clone gene into the patient. So now the gene therapy aims to expand its application, like not only like constricted to the inherited diseases, but to all the other kinds of diseases that have the possibility to be treated by this. Now, if a person is born with a hereditary disease, then the gene therapy can be a corrective therapy for such a disease. Now, gene therapy is a collection of methods that allows correction of a gene defect that has been diagnosed in a child or in an embryo. It doesn't mean that if a person has the disease and now the person is 50 years of age and now the gene therapy can be done. No, it's done at a very earlier stage when the human is just like a child, born and just a very few age in number. And or this can be done for embryo, meaning the human being has not even been born yet. Now here genes are inserted into a person's cell and tissues to treat a disease, right? Whatever the corrective copy of the gene is, that is inserted into the cell or into the tissue to treat that disease. Now, correction of a genetic defect involves delivery of a normal gene into individual or embryo to take over the function and compensate for the non-functional gene. If there is any gene that is non-functional, and because of that non-functional gene, that disease is being caused. So, if we send the functional gene, then the disease will not be caused, isn't it? This is the whole mechanism. Now, the first clinical gene therapy, it was given in the year 1990. This year is very important. It is asked so many times. This gene therapy was firstly given in 1990 to a 4-year-old girl with adenosine deaminase deficiency. ADA is the name of the disease. Okay, adenosine deaminase deficiency. Now, this enzyme, adenosine deaminase, is the name of the enzyme, the deficiency of which is a disease. So, the adenosine deaminase is very important for the immune system so that the proper fighting of the pathogen, fighting of the foreign particle can happen. Okay, but whenever it is absent, so the lymphocyte proliferation is inhibited, meaning the lymphocytes. What are lymphocytes, students? Lymphocytes. Hmm?
T lymphocyte.
What are they? Yeah, T lymphocyte, B lymphocytes are there, you are right. But what is T and B? Like, what are they? Tell me in terms of component of the blood.
White blood cells.
White blood cells, they are
WBC.
WBC, right? Different kind, like they come under the leukocyte, like different types of leukocytes. So, uh, so whenever that happens, so the lymphocyte proliferation is inhibited, meaning lymphocytes cannot divide for the disease, or you can say to combat the disease. So without the T cells, meaning without the T lymphocytes, the ADA deficient children are wide open to the attacks of viruses and bacteria, right? If the person's immune system is like compromised, it doesn't work well, it does not perform what it's supposed to. So then what will happen? Any kind of pathogen can attack onto the human being or the child and cause diseases. So there came another disease at that point of time that was known as SCID, severe combined immunodeficiency disorder, SCID. Now this is caused due to the deletion of gene for adenosine deaminase. So whenever this adenosine deaminase gene, this enzyme gene is inhibited or deleted, then this disease called as SCID is caused. Now, in some children, ADA deficiency can be treated by bone marrow transplantation. You understand what bone marrow is? The mass of cells that are present in between the bone cells, right? That is bone marrow. Now, that is also responsible for the production of the fighter cells, or you can say the lymphocytes, right? So, if the person's bone marrow replacement is done, then the ADA deficient patient can be treated, right? And this, it can be done by, it can be treated by enzyme replacement therapy, ERT, meaning here the injections of the adenosine deaminase enzyme, it is given. So if the person is lacking that enzyme, so the injection of the enzymes are given. But if you think about the bone marrow transplantation, or if you think about constant intravenous injection, so the person or the child has to be taken to the hospital multiple times, isn't it? And if what will happen if we stop the injection? Hmm? The treatment for this disease is the curative injections. What if we stop the injection?
That the person will die due to pathogens and die.
They'll disease, like their disease will not be cured. So it will be back to the zero stage yet again, right? So we can say that these procedures, the normal conventional procedures, they are not curable, right? Then they cannot cure the person completely. So in the case of this thing, the gene therapy that was done using biotechnological processes. So here, lymphocytes from the blood of the patient, they are grown in a culture outside the body, meaning in the lab, the lymphocytes were grown in the laboratory condition. Now, a functional adenosine deaminase cDNA, circular DNA, which is a vector. Okay. So using this DNA, it is introduced into the lymphocytes. Okay, ADA gene is introduced into the lymphocytes. Like in a lab, we are growing the lymphocytes. Now the lymphocyte, because the person who, like from, for example, the patient, from where we took out the lymphocyte, what is that one thing they don't have? They don't have the enzyme called as adenosine deaminase. So now we take that lymphocyte, we are growing it in on a culture plate in the laboratory condition. So if we inject or put the ADA enzyme or gene into the lymphocyte, now if you take these lymphocytes and put it in the patient's back, like body back, so now the lymphocytes will make ADA enzyme and the person's disease can be, you can say, cured sort of. Now, however, as these cells are not immortal, we know that all of the cells have a particular lifespan, isn't it? They die after that. So the patient requires periodic infusion of such genetically engineered lymphocytes. However, if the gene isolated from the marrow cells producing ADA is introduced into the cells at an early embryonic stage, then only it could be a permanent cure. Meaning, when the ADA enzyme or when the gene therapy is done at the embryonic level, meaning much before the organism has been born, then only it can be a permanent cure. But apart from that, all the other methods that we just studied, these are temporary, or you can say periodic treatment that has to be done. It has to be continued all through their life. It is like they're never going to be rid of it. But if it is done at an embryonic level, because you people know that whenever the fetus is developing, a lot of tests are performed so as to check whether it is healthy, whether it is growing fine, whether the organs are being formed nicely, and the gene level also. So if the treatment is done at the gene level while the baby is still inside the mother, while it is a fetus or while it is an embryo, then it can be a permanent cure. Is it clear? Gene therapy is clear to you all?
Yes, ma'am.
Any doubts? Okay, now noted down.
Ma'am, this whole,
Yeah. Yes. This whole thing is important because it's like a very descriptive thing. If you like, if whenever it comes in the exam, you can write this part and you can write this part. But sorry, not the second part. The first part that tells us what gene therapy is. These are like the highlighted points. But if it comes in a 5 marks question, then what you will do? Then you'll write the give whole entire history. Yeah.
yes,
Done, students?
No, ma'am, in the last paragraph. I just started the last paragraph.
Okay, what about the others?
Almost done, ma'am.
Okay. Done, students?
No, ma'am.
Done, ma'am.
Done, ma'am.
All right, students. So we'll keep it till here only for today's class. Whoever has still like some things left, we'll do it in the next class. I'll give you time to take down the notes. Okay. Do you people have any doubts up until now?
No, ma'am.
All right. Okay, students. I'll see you in the next class, meaning tomorrow.